Okay folks, welcome to the first lecture on the human nervous system. The things we need to cover in this lecture are the relationship of the brain and the spinal cord, the structure of the spinal cord, and details of the autonomic nervous system. We also need to cover the ventricular system and cerebrospinal fluid, and then we're going to work on to cover mid and hindbrain structure, function and pathology. In later lectures we will cover the front brain and forebrain structures, and we'll work through it logically like that. So first of all, the brain and spinal cord. We're all aware where the brain and spinal cord are. The diagram here shows a differentiation of your spinal cord. So the butterfly-shaped area in the core is called grey matter. This is divisible when we split this up into dorsal horns and ventral horns, and then you have an intermediate zone in the middle. Dorsal means back, ventral means forward. So as you look at the spinal cord like that, the ventral horn is facing towards your stomach, whereas the dorsal horn is facing out towards your back. Surrounding the grey matter, you have white matter which run straight down, up and down the cord. It's called rostrocaudally. You have a narrow CSF filled channel in the middle. CSF is cerebral spinal fluid, and it's the fluid that circulates through your spinal cord and through and around the brain itself. This tiny little canal is called the spinal canal. So if you have a look at the diagram, on the left you see an undifferentiated brain, so a lower animal hasn't got the same development, versus a differentiated brain further on. So you can see as differentiation increases, the complexity of the spinal column and spinal cord also increases. When we discuss which part of the brain we're talking about, you have to remember that all structures of the brain have evolved from the spinal cord. So effectively, the top end of the spinal cord has formed various enlargements, various growths, and as the spinal cord is folded round to create the structure we would recognise as human brain, we've developed these separate parts. So the forebrain is principally involved with things like olfaction, so your sense of smell, your sense of taste. The midbrain deals with vision. Hindbrain deals with vibration, so hearing. These are the sort of key senses that you have as a human being. I want to show you a bit more detail on the spinal cord as it is so important. So here we've got the spinal cord, you can see the grey matter in its classic butterfly shape. The white matter is forming chunks that go ventrally and dorsally to the grey matter. At the top, you're effectively looking as if somebody is lying on their front here, so the dorsal root and the dorsal side of your spinal cord is facing upwards, and that would be towards your back. So sensory information from a sensory receptor, for example in your skin, passes up through the dorsal root cells and into the dorsal root ganglion. Once in there, it then passes up through the dorsal root, so this could be any spinal nerve. Motor commands leave via the ventral roots. So in effect, the signal goes in through the back of the spinal cord and out through the front. The grey matter itself is composed of cell bodies, whereas the white matter is composed of axonal tracts, and it's the myelin sheath that gives it this white appearance, hence why it's called white matter. Discussing that further and looking at how we carry input to and from the brain. Here again is a slice of the spinal cord with the dorsal end closest to the top of the slide and the ventral end at the bottom. Dorsal columns carry ascending sensory input to the brain. These are the afferent nerves. So in this case we have an alpha-beta axon sending information through the dorsal root, the dorsal root ganglion, and into the grey matter and the afferent tract there. So here's the dorsal column at the top, which is the white matter. That's the bodies of the nerves. That's where your sensory input is passing up. Descending or efferent spinal tracts carry motor commands from the brain to the muscles via the spinal cord. So you have two-way traffic going on in the spinal cord. As long as you remember that grey matter is cell bodies, white matter basically the sheaths and the axons of the neurons, you won't go far wrong. When we look at the spine itself, so in this diagram you can see roughly how the spine is fitting through the vertebrae. So you have the spinal cord down the centre and the dorsal roots coming off that. Dorsal root ganglia are the bulges that we see as it comes out of the spine to the vertebrae, and that's where you have major cell bodies forming, so you have this lump. The ventral root is at the front of it. You have various connective tissue sheaths which protect the spinal cord itself before it's fully protected by the spinal processes of the vertebrae themselves. If you move around to the front, you see where the vertebrae are identified and the intervertebral discs, the spinal discs, are identified. You then have spinal nerves coming off through these spinal processes. The next line shows the sympathetic trunk and the sympathetic chain ganglion. Again, this is where you have cell bodies coming down through the sympathetic nerves. The autonomic nervous system controls all your visceral functions, so it controls things like your heart rate and your breathing rate. You have sympathetic or excitatory and parasympathetic inhibitory divisions. Remember what I said in the previous lecture on the cardiovascular system. If you remember para as being parachute, so it comes down, the parasympathetic nervous system calms everything down and is inhibitory. Together, these two parts of the nervous system control all of your visceral functions, so they control everything that you need to maintain homeostasis. When we're thinking about the autonomic nervous system, you have to remember that you have somatic and visceral branches. So somatic relates to something called somites. When you're actually in development and your nervous system is developing, we have segments forming, and these are the somites. This controls the voluntary part of your nervous system, such as voluntary movement, skeletal movement. You can control your breathing rate to a point. However, if you hold your breath for long enough, you will pass out, but as soon as you do that, the autonomic system kicks in and starts to control your breathing rate for you. The visceral branch relates to the internal organs or the viscera, and this is entirely involuntary. So the movement of your intestines, of your stomach, is entirely involuntary. You don't have to think about it. Looking at the diagram, you have the central nervous system at the top, consists of the spinal cord and the brain. There's virtually constant interplay between these two structures throughout the body. Information goes backwards and forwards to the brain via the spinal cord. Motor or efferent neurons come out of the spinal cord and into the peripheral nervous system, where they then split into the somatic system to innervate skeletal muscle and the autonomic system, which then splits further to the sympathetic system and the parasympathetic system. Together these innervate smooth and cardiac muscles and deal with the glands, so pancreatic secretion, pituitary gland, everything like that. On the other side you have internal and external stimuli. So internal stimuli could be a change in blood pressure from baroreceptors, a change in blood chemistry, so an increase in CO2 concentration. External stimuli could be cold feeling on your skin or an increase in pressure. The sensory receptors, all except those for vision, then travel up through the sensory and afferent neurons into the brain itself. That then results in efferent neurons signalling through the spinal cord and back into the peripheral nervous system. Your central nervous system has three potential neural outputs. Looking at this diagram straight away, we've got the central nervous system at the top, peripheral nervous system in the middle, and then effector organs at the bottom. The three potential outputs are the somatic motor and then the autonomic nervous system, which is split into sympathetic and parasympathetic. The sole output for the somatic motor system is the lower motor neurons of the ventral horn, the spinal cord and the brain stem, so the final common pathway for the generation of behaviour. These nerves come straight down through the peripheral nervous system as a somatic motor fibre and innervate skeletal muscle directly. Some activities, such as salivation, sweating for example, depend on the autonomic nervous system, and these visceral motor responses are affected by whether it is a sympathetic or parasympathetic division of the autonomic nervous system which has been stimulated. In this case, the ganglia for these neurons lie outside the central nervous system, so these are the lower motor neurons are the postganglionic neurons. For the sympathetic nervous system, the preganglionic fibre is short, The autonomic sympathetic ganglion is located in the peripheral nervous system, and they then have long postganglionic fibres before innervating their target organ, which could be smooth muscle, cardiac muscle, or the gland cells. Conversely, the parasympathetic nerves have very long preganglionic fibres. Again, their parasympathetic autonomic ganglion is located in the peripheral nervous system. Their postganglionic fibres are a lot shorter than the sympathetic nerves, and again, they then innervate things like smooth muscle, cardiac muscle, or the gland cells. I mentioned cerebrospinal fluid, or CSF, right at the beginning of the lecture. What you must remember is that your brain and spinal cord are hollow. They're covered, the brain, with something called the arachnoid membranes. These are the meninges. We have lots of cavities in the brain that are filled with cerebrospinal fluid. So if you look at the diagram of the brain, first of all, on the left, in blue, you can see the human ventricular system. So these are the ventricles of the brain. Don't get these mixed up with the ventricles of the heart. They are completely different. As the brain has developed and grown, these ventricles have become distorted and peculiar shapes. However, the basic relationship to the brain of the ventricles is exactly the same in a complex brain versus a less complex brain. So, coming up through the spinal cord, you can see the spinal canal highlighted in blue, coming up behind the cerebellum and up into the brain itself. Here you have the lateral ventricles are the two at the top that seem to be large, almost like butterfly wings. Third ventricle is in the middle, and the fourth ventricle is lower in the brain, just above the brainstem. If you look over to the right-hand diagram, you can see the brain tissue itself at the bottom, then large arteries coming through, innervating the tissue. The first membrane is called the pia mater. It's quite a delicate membrane, although it is fairly tough. It's not a very thick membrane. You then have the subarachnoid space before the arachnoid membrane starts. You then have the subdural space, and the dura mater is the final membrane that covers the brain. The dura mater is exceptionally strong. It's very, very tough, very thick. It's very difficult to actually damage it, but that is pretty much all that lies between your brain surface and your inside of your skull. All of these various cavities, such as the subarachnoid space, the ventricles, have got the CSF inside them, and that CSF is flowing between the ventricles, the various spaces of the brain, and the spinal cord at all times in a constant flow. So this takes us on now to actually working through the sections of the brain. So we're actually going to start at the hindbrain first. The reason for this is because as the spinal cord has developed, and as it started to develop these extra differentiated structures, the hindbrain was the first bit to develop. Effectively, if you straightened out the brain, the hindbrain would be that first bulge to come out from the spinal cord. What's actually happened in humans is our brains have become so large and so differentiated. It's effectively grown up and flipped back on itself and then back over again to give us the classic brain shape with the spinal cord coming down from sort of the rear middle. The medulla is the most caudal section and is the least modified from the spinal cord. If you look on the diagram, you have the forebrain, midbrain and the hindbrain. Depending on the level of differentiation, you may see different structures. So the bottom part of the diagram, if you look first of all at the most differentiated side, you see that you've got that fourth ventricle at the top. You then have the medulla itself, and underneath you have two medullary pyramids. The medulla controls your autonomic homeostatic systems, so damage to this part of the brain is fatal. It controls your breathing rhythm, your temperature control, and maintenance of blood glucose levels. It includes something called the reticular formation, which we'll come on to later as needed. Moving forward from the hindbrain, we have the pons. The pons is the rostral hindbrain. So if you have a look at the diagram here, this is showing how the rostral hindbrain actually differentiates into the cerebellum and the pons. The cerebellum is formed by the growth and fusion of the rhombic lips, which are the parts of the brain that actually come over. So if you look down at the lower part of the diagram, the middle part, effectively, is showing a sort of mid-differentiation stage. So you have the pons underneath, and as those two rhombic globes actually come and form over, first of all, the fourth ventricle is made much smaller, but you've developed this large structure which will form the cerebellum. I say the fourth ventricle is a CSF-filled space, and again it's at the core of the hindbrain. So you have the rostral hindbrain with the dorsal cerebellum. Remember, the dorsal means back, ventral means front. So here you've got the dorsal cerebellum is pointing out towards the back of your body. You then have the ventral pons in front of it, which bridges the left and the right sides. The pons carries axons into the cerebellum and contains important nuclei which we use in controlling sleep, waking and arousal cycles. So how we wake up and the understanding of how our brain actually controls that. If you want to look a little closer at the hindbrain for the cerebellum, remember this is the strange structure that's formed at the back. If you look at the brain at the top, you can roughly work out how the brain structures have curved round, which gives you these strange structures, these lobules at the back forming the cerebellum. When we look at the cerebellum itself, it's split into various sections. So you have two hemispheres, and that central point is called the vermis. Because it was the rostral lips that came over and fused to form the cerebellum, it is not in two separate hemispheres like the rest of the brain, but is one complete structure. The folds in it are called folia. At the bottom of the diagram you can see where the medulla lies and the spinal cord. If you look over to part C of the diagram, they actually slice through the cerebellum here, so you can see the fourth ventricle in the centre, you can see the pontine nuclei in the pons at the front, and you can see deep cerebellar nuclei which show as a different colour within the tissue. The cerebellar cortex is highly folded. The cerebellum integrates information about body position with the motor commands, so it tells your body where the limbs are, for example, in relation to your head, or where they are in relation to the rest of you. So it's that feeling, you know, when you get out of bed, you put your feet down first because you know that is the bit that goes first, and then you stand up. If you tried to do it with your hands, you'd fall over. The cerebellum learns movement sequences and will subconsciously run them. As you develop from infancy into adulthood, everybody knows, seen that picture of the child learning to walk. It's because these movement sequences are being structured in the brain. Once they're learned, they become subconscious. If you damage this part of the brain, you lose a lot of your coordination. So you'd find it very difficult, for example, to lift a mug in your hand straight to your mouth. You'd find that you wouldn't be able to aim very well. The midbrain is a bit of a strange structure. It actually differentiates into two parts called the tectum and the tegmentum. The CSF-filled space in the middle here is now called the cerebral aqueduct. So if you look again at the most differentiated part, you can see the tectum at the top, and then the tegmentum is the middle bit. It's a very simple structure, so the roof is the tectum and the floor is the tegmentum. Ultimately, it will differentiate into parts of the brain that deal with visual tracking, called the superior colliculi, and inferior colliculi, which deal with auditory processing. So this part of your brain deals with how your eyes follow a moving object and how your ears actually process sound. This aqueduct connects ventricles 3 and 4 throughout the brain. So you have lots and lots of connections all the way through that you may not think of. The midbrain is extremely important for movement. In this point, you have two pairs of dopamine nuclei in the midbrain. You don't need to worry too much about the names of the various structures up to this point. You need to know the major structures of the brain, such as the hindbrain, pons, cerebellum, medulla. Don't need you to worry too much about the tectum and tegmentum at this point, but I do need you to understand this. So you have these two nuclei in the brain that produce a lot of dopamine. If you look at the diagram first of all, you can see the ventral tegmental area, and you see these two structures. The blue arrows coming off are showing you where those nerves are going that secrete dopamine. So you've got a fairly concentrated proportion of the dopamine system in the frontal lobe and the centre of the brain up to something called the striatum. These two nuclei are the substantia nigra, which leads to part of the control of the motor system and has the output to the striatum, which is the basal ganglia, and the second dopamine nuclei is the ventral tegmental area. Degeneration in these parts of the brain leads to Parkinson's disease, which leads to tremor and difficulty initiating movement. It's not actually affecting the cerebellum as such, but it's affecting these dopamine nuclei and the dopamine signalling system throughout the brain. The midbrain is also involved with our pleasure and perception pathways. So that ventral tegmental area, that second dopamine nuclei, projects to the limbic system and the frontal cortex. The mesolimbic pathway terminates in the nucleus accumbens, which signals reward and is a main activity site for addictive drugs. The top diagram here shows a human brain, whereas the bottom diagram shows a parasagittal section of rat brain, so much more simple. If you look at the rat brain for the moment, from the right you can see the nerve, the spinal cord coming in as a cerebellum, and then going through to the midbrain, the forebrain. So here you have the dopamine neurons of the ventral tegmental area, shown in blue, which have synapses with the nucleus accumbens. So addictive drugs such as cocaine have an effect on strengthening the pathways up in the nucleus accumbens. Heroin and nicotine directly affect the dopamine neurons in the ventral tegmental area. As a result, when you take those substances, it signals the reward pathway, which is why they become highly addictive. If you have malfunction of the mesocortical pathway, we see it's quite common to have this in schizophrenia, although we don't actually understand the mechanisms behind that. In summary, the relationship of the brain and spinal cord has been discussed in relating to the structure of the spinal cord itself and your autonomic nervous system. From this lecture, you need to understand about the ventricle system and how CSF is actually distributed around the brain, and I need you to understand about the hind and midbrain structure, their functions, and any key pathologies such as addictive drugs and Parkinson's are actually influenced by the structure of the brain and what happens in these conditions. In the next lectures we'll come on to discuss other parts of the brain. In the meantime, if you're struggling with anything, as always, please get in touch.